So the place where we left off was looking at glomerular filtration as the first of the 3 steps that are involved with the formation of urine. So we looked at the barometer reflex. That's where we change the afferent arterial to vary or fine-tune our glomerular filtration rate. The other thing that we want to think about with glomerular filtration is not so much part of the actual process that's happening at your filtration membranes. Remember, the filtration membrane had 3 layers to it. Put that up here. But we usually talk about it when we talk about the glomerulus because it's anatomically close to this area of the nephron. And so this is going to be our renin, angiotensin, aldosterone system. And so the part of the glomerulus, this is my vascular pathway, and then I've got the tubular pathway here. So Bowman's capsule, proximal tubule, loop of Henle, distal tubule, and collecting tubule. So in this area of the nephron, we have our juxtaglomerular apparatus. And this is going to have apparatus. This is going to have sensory cells in it. That's an A right there. That are going to detect things like our sodium levels, sodium concentration, and also detect pressure. And the pressure is created by the amount of blood in the afferent arteriole, as well as the amount of filtrate in our tubular pathway, because notice that the tubular pathway is also tied up in this region. So the juxtaglomerular apparatus is in a great place to pick up on sodium concentration as well as how much pressure is being exerted against it. And this is going to be a direct result of how much fluid you've got in your tubular pathway. So anyway, the juxtaglomerular apparatus, if it detects a drop in your sodium, so we'll do a down arrow for the drop in sodium, we know that a drop in sodium means that we also have less solute pooling water. And so a drop in sodium is also going to indicate that we're getting low in the amount of fluid that we have. So these two things go hand in hand because water follows the solute. So we release our enzyme, renin, okay, and renin is going to go into the bloodstream and act on angiotensinogen. And angiotensinogen is put into the blood by the liver. So it's a great example of, I'll put from the liver into the blood. It's a great example of a plasma protein, one of the many plasma proteins that the liver produces. So we're going to activate our angiotensinogen, and once it's activated, it becomes angiotensin 1. And it's still going through the circulatory system, so it goes through the pulmonary capillaries, and in the pulmonary capillaries, it's converted to angiotensin 2 by our angiotensin converting enzyme. Okay, so that's what the ACE stands for, and of course you need to know that that's happening in the capillaries of the lungs. Then angiotensin 2 goes to the adrenal cortex and tells it to release aldosterone. Okay, so that's our pathway part. Now, the aldosterone is going to go to our nephron, specifically the distal convoluted tubule and the collecting tubule. So kind of the last parts of our tubular pathway. Okay, so our filtrate is going through the tubular pathway and lining these tubules. You've got simple cuboidal cells, and this would be the luminal membrane on the left of the cell, and over here on the right we'll make this our basolateral membrane. And so the aldosterone is going to come over here and tell these cells a couple things. It's going to tell them to increase the activity of their sodium-potassium pumps. And remember, the sodium-potassium pumps are on this basolateral side. They're not on the luminal, they're on the basolateral side. And that's going to push sodium out of the cell. And so we ramp up our activity of these sodium-potassium pumps. And when we do that, we're going to decrease the intracellular concentration of sodium. So we're pushing it out, and so that's going to even further drop this intracellular sodium concentration. Now, what that does is it's going to make a bigger gradient between the filtrate and the inside of the cell. So aldosterone is going to ramp up sodium potassium pumps, thereby lowering our intracellular concentration of sodium, but it's also going to increase the number of sodium leak channels on the luminal membrane. So make sure you know where both of these things are happening. Remember, the sodium-potassium pumps, these are on our basolateral membrane. The leak channels are going to be over here on the luminal membrane. And it's a luminal membrane because it faces the lumen of the tubule where the filtrate is. So this is the luminal membrane. Okay. So we increase the number of these sodium leak channels, and this is going to increase our gradient, and based on what we know about diffusion, this is going to increase the rate at which sodium diffuses into our cell, okay, from the filtrate into the cell. And as that happens, we've also ramped up that pump so we can get it out of the cell just as quickly, right? Now that's going to help us reabsorb our sodium, because once we've pushed out that sodium across the basolateral membrane, remember, you've got your peritubular capillaries over here. I go back to my previous page, because remember, this little cell that we're looking at, this is in the distal tubule and the collecting tubule. So it's in the last part of our nephron, okay, back in here. And so remember, our peritubular capillaries, the squiggly line, is going to be surrounding these tubules, and it's going to be picking up the sodium, the sodium that's reabsorbed, all right? So the sodium doesn't just like stick around necessarily. We're reabsorbing it and putting it back into the bloodstream, where it's going to help us pull water back into the bloodstream, right? So that summarizes your renin-angiotensin-aldosterone pathway, and the place in the book that talks about that is on page 393 and 394, right?